Good morning, Professor Vincent. Thank you for joining the World Federation Neurology YouTube channel. For our viewers, Professor Angela Vincent doesn't need any introduction. She is definitely the queen in autoimmune neurology, an illustrated career expanding over 4 decades, and she is basically responsible for most of the autoimmune antibodies that you naturally request these days in your emergency department, specifically voltage-gated potassium channel antibodies and some of the other antibodies. If she's not directly responsible, she's part of the gang which is responsible for these things. So she really changed the atmosphere or landscape of how we look into these disorders. Professor Vincent, thank you for joining us again.
And thank you. It's a pleasure to be interviewed, particularly from Australia. It seems quite remote.
I understand. That's the power of technology these days, isn't it? Professor Vincent is very generous supporting your organization, World Federation Neurology, to introduce inaugural Angela Vincent Award, which you all know that one of our colleagues from Chad was fortunate enough to win. We thought we would talk to Professor Vincent about her career, as she herself has a very inspiring story. Professor Vincent, you have an illustrated career. It has a unique combination of translational neuroscience, expanding over several decades, yet you have been happily married and you are a mother of 4 children, as I gather. So tell us a little bit about your early life and how this journey began.
Well, it's always difficult to say. I didn't plan any of it, or at least hardly. I think I was always interested in science as a child, but my parents weren't specifically scientific, so that, and neither were any of my siblings actually. So I probably got it from an uncle who actually was a clinician scientist, but I didn't see that much of him, but I think I probably have some sort of genetic trait there. And I was curious about nature and did a few little experiments, nothing of any note, and really just wanted to study science. So as I got into my teens, I would start reading lots of books about the brain and physiology, anything really that I could try to understand. And I insisted on doing science at A levels at school, which was not that common in those days for women. So I did get into medical school, but I have to say that I was not a high achiever at any stage of my career up to the last few years, perhaps. I certainly wasn't a high achiever at school. I was, in the top 15% perhaps, but nothing spectacular. So it was all rather serendipitous.
Brilliant. So tell us about your early days in the medical school. Did you keep your interest and enthusiasm in brain and brain-related disorders?
No, I wasn't particularly entranced by the neurologists, actually. I won't tell you who they were. I mean, neurology in those days was a rather dull subject. It was all about sort of clinical acumen, yes, and studying the patients very carefully. But at the end of the day, it always seemed to end up the clinician saying,
doing nothing.
Well, I've seen this before, therefore it must be such and such. But there was no science in their thinking, at least very little in the medical school I went to.
And so, there was probably very little therapeutics also at that time, wasn't it?
Absolutely. And really, there were other things that were more interesting at that time in science, which was more the biochemistry of living cells and how DNA replicated, and all of that was beginning to really get understood, and that was very exciting. So the brain per se was a bit of a black box, and I remember our neurology or brain lectures during the early years, and it was just a matter of sort of learning by rote various things, but nobody actually made you believe that you could understand it.
It was a different era compared to now, I guess,
It
as
was
well.
actually, it was 60 years ago, so, or at least almost 60 years ago.
There was hardly any neuroimaging. Stroke, there was absolutely no treatment, no autoimmunity at all. You make a brilliant diagnosis, wash your hands and walk away, and patient probably die.
And if they hadn't seen a patient like that before, or they hadn't read about it in some old textbook, then they actually just washed their hands and walked away anyway, without a diagnosis. They were not curious in the way that I thought they ought to be, at least the ones I knew. Now, that doesn't mean they weren't all like that, because I know some of them were very curious.
Fortunately, that didn't deter your curiosity. You kept your enthusiasm in curiosity going at a faster rate.
Yes, I mean, I did my house jobs, which is the first year of internship, which enabled you to register as a doctor. Mm-hmm. And I did those, and I actually enjoyed them because I'm good with my hands. And so I enjoyed the hands-on aspects of medicine, actually doing things. I did some appendicectomies in my surgical job. I enjoyed it, but it wasn't what I wanted to do. And so I went and did a master's in biochemistry for 2 years at University College London.
Mm-hmm. So you basically told me that you fell in love with biochemistry even during undergraduate years. You were mesmerized with DNA replication and so on.
Well, yes, it was just a very fascinating time, and I was lucky to do the master's when I did do it in a environment in University College, which was very active and very encouraging. There weren't that many women around, but I seemed to have perfectly good relationships with the senior people, and I never found that a problem.
So that would have been somewhat peculiar. So you finished the internship. So everyone else would have gone on to get senior house officer jobs and so on, which probably would have brought them a bit more money. But you opted to go and do a master's in biochemistry.
Yes.
And
sorry for interrupting, but at that stage I wanted to be a basic scientist.
Mm-hmm. And I believe
medicine.
I believe your initial experiments were not that successful. Tell us about how disappointing they were, and how did the table turn?
Well, I mean, there were two aspects to that. In the master's, I did a computer project, which I think is worth relating, because I was actually having to stay at home because my childcare had fallen through and was no longer available. So I had to stay at home with my little daughter, who was about 1 time. And the best thing to do as a project, therefore, was something which I could do from home. So I did this computer simulation, which was amazing because I was using this IBM 360, which was the room in which it was held was about as large as the sort of average institute coffee room.
Right.
It was enormous. And you had to punch cards every morning to put in new data. And then when it was printed out the following morning, you could see whether it worked. And of course, it never did to begin with. And you had to try again with new punch cards. It was an extraordinary thing to do, but I'm very pleased that I did that. So that was my master's project. Mm-hmm. And then I was offered 2 jobs, actually. One of them was working for somebody and doing a PhD, but I decided I had to try and earn a little bit of money. Mm-hmm. So I was a research assistant. Mm-hmm. And I needed the money for the childcare because, of course, one knows how expensive that can be. And I had a project for 3 years, which was spectacularly unsuccessful. I had to try and separate pre- and post-synaptic membranes from brain tissue. Mm-hmm. The idea was to find out where cyclic AMP was active, mm-hmm. or what in fact is the kinase, in fact, as it turns out. But that was a very new discovery, and they wanted to know whether it was on the presynaptic side or the postsynaptic. And I was supposed to be separating these 2 membranes. And of course, it's a very difficult thing to do, and I don't think anybody's done it yet. Mm-hmm. Maybe they don't need to now. But I spent 3 years looking down an electron microscope, trying to decide whether I had achieved a separation with the various enzymes I tried to solubilize the gap in between them. And it was pretty soul destroying, but I tried a few additional tricks and I began to learn to think like a scientist. And I think that's a very important thing.
Explain that more. This is something the significantly lacking in current generation of clinician students in particular, even among some of the younger neurologists. So explain this a bit more, scientific thinking.
Oh, I don't know. I think it's for me. I think it's different for other people, but for me, it's a matter of visualizing what I'm trying to do and trying to visualize it often at a molecular level, which wasn't so easy in those days because, of course, now we know much more about the molecules. But it's also seeing what you have in your test tube and what's going on. And I actually often encourage people to draw, even if they're no good at drawing, draw their experiments. Because I think that a combination of using your hands, thinking and visualizing what you're doing helps your brain come up with different ideas.
Why certain things are happening in certain way.
Yes, and what you might have missed, perhaps, in your original thinking, and what you might try to do differently the next time you do an experiment. But it's a slow process. It's not something that comes, in flashes, at least not in my case.
I also believe that you had a particular supervisor, a particular senior scientist in the department who were supportive of your bold thinking at that point of time of your career. I believe that was during your third year of your research year. Is that correct?
Sorry, I'm not quite sure what you're talking about.
While the experiments were failing, you went ahead and questioned one of your senior scientists with a very bold idea of doing things differently.
Oh well, I think no, that's not quite what we want to say here. I think we can edit this bit out. When I was doing this work, I just began to wonder what I was going to do next, because it obviously wasn't going to lead on to anything. So I was wondering who to work with, but I did know that one of the things I would like to be able to do would be to label the pre or post-synaptic membranes in a way that I could measure independently of the electron microscope. And at that time, a very nice, useful snake toxin called bungarotoxin had just been reported to bind very well to the acetylcholine receptors at the neuromuscular junction. And at that time, we really didn't know that it wouldn't necessarily bind well within the brain, because people just did not know enough about brain synapses, which seemed extraordinary. Anyway, the person who'd done the work on bungarotoxin was Riccardo Milady, who worked literally on the floor above me. And I knew of him from, chance encounters in the lift. And I knew he was a nice person. So I went and asked him if I could come and work with him and perhaps try his toxin on my preparations. So if you think that's bold, it wasn't bold, but it was lucky.
Yes, that was what I meant being bold. But you like to categorize it as lucky and serendipity again.
Well, it wasn't bold because of the kind of person he was. So despite his distinction, you would never have thought so coming across him in the lift. He was a wonderful person to work with. And I had 5 years working with him, during which the beginning of my career really did begin to come together in a way which, of course, I've been very fortunate.
By this time, you would have had 2 or 3 children now?
I had a second during my master's. No, sorry, during the research that I did so unsuccessfully. And then during the time I spent with Riccardo, I had the third, yes.
Right. So tell us about the neuromuscular junction and brain. How did your career expand from then on?
Well, in fact, I didn't do any of the brain experiments because once I went to work with Ricardo, we did work only on the neuromuscular junction. And that was, of course, the most important thing in those days, because it turned out that myasthenia gravis was a disease of the neuromuscular junction, which we knew, but we could now investigate it because we had this toxin as a probe for the acetylcholine receptors. And just for your audience, it might be important to say that at that time, I knew people, well, at least one, at University College, who did not even accept that acetylcholine bound to a protein receptor. Some people, acetylcholine just caused a change in the membrane without actually binding to anything very specific. Receptors were a very new idea at that stage, and the acetylcholine receptor from the neuromuscular junction was the first to be purified. Subsequently, it was the first to be sequenced of the membrane receptors. So it was incredibly lucky to be in that field at that time. And then with the association with a human disease that turned out to be very treatable, that was an added bonus.
It is indeed. I mean, for the audience, we should remind them that every time when they request acetylcholine receptor antibodies now with ocular myasthenia or generalized myasthenia, at that time, there were clinician scientists or clinicians who did not believe even that there was a thing called acetylcholine receptor.
Anyway, that changed pretty soon because Ricardo had purified them and had shown that it was a protein and that it had 4 subunits on Western blots, etc., etc. So that was just an interesting factor that until one could purify these receptors, it was not really clear that they were there.
So now to a different question, Angela. When did you publish your first paper?
Well, actually, the first paper was the computer simulation, which I did, which was looking at just simulating the changes in ATP and phosphate in muscle during activity. And I knew very little about the basis of this project. I just enjoyed doing the actual computing, which was really good fun. At that time, I would quite like to have gone into computing medicine, actually. But early, I think, for that. Anyway, so that was my first paper, and then after that, it took quite a while because I got nothing out of my 3 years of unsuccessful electron microscopy. And then when I went to work with Ricardo, it was a little bit of a gap, I think in about 1975. So probably another 5 years before I actually published at all with him. But, you know, then we published a few papers and then it began to
flourish.
Well, it began to as soon as one gets into the clinical arena, it's usually easier to publish papers, to be honest.
I think this is something that we should remind our viewers and audience. Just because you may have a hiatus for 1 or 2 or 3 years is not the end of the world. You just need to persevere with your dreams and keep going.
Absolutely.
Especially keep feeding the curiosity, isn't it? Which is what you have been doing throughout your career by the look of things.
Well, it's lucky if you have the opportunities to do that. And of course, that can be quite difficult with today's funding being very kind of systematic, not giving you a lot of flexibility. And I think one of the lucky things was that when we started working on the clinical disease, because it was all very new and exciting, to have an antibody to be able to show that patients got better with treatments that reduced the antibody levels, and that the thymus was involved. I mean, everything had been there before, but suddenly it became much more interesting because one began to understand the mechanism. And that meant that it was easier to get long-term funding, which supported you, beyond the experiments that you had actually planned to do. Gave you much more flexibility.
Tell us about the story of voltage-gated potassium channel antibodies. How did they come about?
Well, we had by this time we were in Oxford. So after Ricardo Milady, I went to work with John Newton Davis at the Royal Free. John was also a neurologist at Queen Square, of course, but the labs that we worked in were at the Royal Free Hospital. And that was for 10 years, actually, and then we moved to Oxford. During the 10 years at the Royal Free, we helped to discover the voltagegated calcium channel antibodies in the Lambert-Eaton syndrome, working with Dennis Reyes, electrophysiologist, and many other collaborators. And then by the time we got to Oxford, John had already noticed that some of the patients that he saw with thymoma had neuromyotonia, or Isaac's disease, as it was often called. And he also noticed that there was some hint of autoimmunity in their histories. They had a thymic tumor in some cases, and although many of them didn't have a thymic tumor, a few of them had myasthenia as well as neuromyotonia. So it was fairly obvious from that it was likely that neuromyotonia was an autoimmune disease, at least in some. So what was the target? Well, we'd already worked on the calcium channels, which are presynaptic. And obviously, in the Lambert-Eaton syndrome, you have a reduced release of acetylcholine. Probably in neuromyotonia, where you have this hyperexcitability of the nerve, there must be an increased release of acetylcholine. And that's what we now believe. The question is, what would the target be? And one of the obvious targets was a potassium channel, because most people know that if you inhibit potassium channels, you get extra hyperactivity of nerves, or indeed brain neurons. So we looked for antibodies to potassium channels. And we did this partly with colleagues around the country. Ian Hart in Liverpool, for instance, was very much involved initially. And eventually we found that if we used dendrotoxin, another toxin, to label potassium channels from brain tissue, we could show that some of the neuromyotonia patients had antibodies that precipitated these potassium channel complexes. Now, the trouble was that we were actually slightly wrong, because it wasn't the potassium channel that was the target of the antibodies. It was actually other proteins that were complexed with it in the brain tissue that we were using for these experiments. And that, I like to quote a quotation from, God, now I'm going to forget the name of the person, which is very like me, I'm afraid. But the fact is, it's very useful. It's very nice to be right. It is much more useful to be wrong. I think that's a wonderful.
It is a wonderful quote, yes.
Holmes. So, and it's a wonderful quotation because it's absolutely correct. The fact is, if you're wrong, and if you try to find out why you were wrong, you may find things which you hadn't expected. And that's exactly what happened to us, because we found that the proteins that, or two proteins that were complex with the potassium channels were Caspar-2 and LGI1. And we now know that those are the targets for the antibodies that precipitate the whole complex, including the potassium channel. And that Caspar-2 antibodies and LGI1 antibodies are present in patients who can have a lot of overlap, but quite often are fairly distinctive clinical features. And that was a very interesting and satisfying project, because to actually work something out properly from basic principles took a little bit of ingenuity and a bit of good fortune, as usual.
It certainly changed the game. I remember seeing my first patient with LG antibodies during a post-tech round. There was this lady who was having these occasional funny turns, which, as you know, people call as brachiofacial seizures. And I somehow read some of your papers by that time. I believe that was the second case reported in Australia a few years back. And I told my registrar that this must be that Angela Vincent group's antibody. Let's test this specific antibody to see our luck. And it was indeed, and patient got better eventually.
Yes, well, that's always very nice to hear, I must say.
That's from the bedside.
Yes.
We'll change the tack a little bit. We do know that you have a brilliant, amazing career, and you published well, and you are well regarded in this field worldwide. To the extent that you won a large number of awards, because this is World Federation Neurology YouTube channel. I have to remind viewers that you were one of our medal recipients during the last World Congress. Congratulations on that too. When did you involve with World Federation Neurology for the first time? Can you remind us?
Well, I think I was just invited to speak at various meetings of the World Federation, and quite often doing teaching on the neuromuscular junction, or on channel diseases, those sort of aspects. More recently, I've sometimes talked on or contributed to teaching on neuromyelitis optica. We, of course, discovered the aquaporin 4 antibody. That was by Van der Leen. But on the other hand, Oxford's been very active in trying to improve assays for all of these antibodies and to make them available to as many people as possible for their diagnosis. And so the Mayo Clinic and Oxford have a strong relationship now, often collaborating to try and help establish the best assays and to make sure they're well recognized.
Definitely. So the changing the tack again, thanking you for sponsoring this award. As I said, it has gone to a remarkable person in Chad, South Africa, that we would all watch how he would grow from strength to strength, and how this would transpire to better brain health and better care for Chadians and Africans in general. Why did you wanted to sponsor this award? What was the reason behind sponsoring this award?
It wasn't really my idea to sponsor an award as such, and I feel quite embarrassed about it being called Angela Vincent, because all that happened was that when I was lucky enough and really absolutely thrilled to receive the WFN Clinical Science Award in Kyoto in 2017, I was a little bit surprised to be given a check. And I can't remember exactly what the sum was, but it was $5,000, I suspect, or maybe, yes, I suspect it was dollars, but I have no idea now, because I just gave it back and said, look, I don't really need this. Why don't you use it to help some people, bursaries for people from low and middle income countries? That's really what I wanted to support. Because one of the things I've always admired about the WCNs that I've been to, the congresses, is that there are a wide variety of people from different continents, and particularly since Marrakesh, when I think that really exploded into the African continent, and that was wonderful to see there. And I've always been quite keen on supporting and helping people in whatever way I can from less privileged countries. So that seemed to me to be the best use of the money. And I just left it at that. And then I got an email a few weeks later from Raj Shakir, who was the outgoing president, and he said they wanted to call it the Angela Vincent Award. And I really was not very happy with that. But they went ahead and did it anyways. I'm actually now, well, flattered for it to be called an award, as a sort of formal award. I'm delighted that Dr. Sakadi has got it, and he's exactly the kind of person I would have hoped to get it. And I just hope that other people might consider also helping the World Federation by giving named awards. If this is going to help other people give awards that can be used in a similar manner, then hopefully eventually it will just be called international awards for the WFN. But if I've helped to sort of get that started, that would be very good.
I think you hit the nail on the head on various fronts in that answer, Angela. I have a special quote to most of my trainees and medical students. I believe this world has enough resources for us to sort out most problems that we need, at least in brain health related matters. And there are enough of us around who are happy to break the barriers between countries. You are in Oxford now, and I'm doing this for World Federation of Neurology. I'm at my home in Melbourne, and we are talking about Chad in South Africa. You can see how the technology has cut across all these continental barriers. And the other thing is, yes, we must help and support our colleagues who are struggling to make ends meet for their patients and their trainees and so on. So thank you for that, and let's hope that some of the other more privileged viewers of this would think of supporting World Federation Neurology to do exactly the same, to the approach more countries. Changing the topic again, share a few things about your personal hobbies and some of the things that you're happy to talk about. The things that you're not happy to talk about, you don't have to worry about. What do you do during your spare time?
Well, I sort of given up spare time. It's difficult when you're a woman, obviously. I know men are now helping a lot with the families, but I think still the woman has the major role in bringing the children up. I sometimes think I just trod water when my children were growing up, because career-wise, I wasn't trying to get ahead. I was happy. I was lucky to be in a position where I didn't have to fight for grants too much, because we were very fortunately well funded. So that was fine, but of course it wasn't the future really. And as soon as they'd left, or more or less all left home, I was able to kind of somehow take off, and that was very important for me. And since then, I really haven't had much spare time anyway. So I don't do a lot, but I love opera, and particularly Wagner. Mm-hmm. I used to play tennis when I was young, and I was quite good at it. So a few years ago, I started playing tennis again, just once a week with a coach. Mm-hmm. And we just, I just said, all I want you to do is get me running around the court and see if I can still play. And I sort of could, which is very reassuring, and I enjoyed that. But mostly it's the children and the grandchildren. I have 3 grandchildren now, and they're grown up. 2 of them are teenagers. So there's enough to keep me busy one way and the other.
I'm sure you get enough academic activities all over the world also to keep yourself busy. At the moment, you supervise a couple of PhD students also?
Well, I've only got 1 left now. One of them left about a year ago. I'm not really supposed to take on new ones, at least not in Oxford, but I have a few connections now in London where I'm helping to supervise a few people and helping to, I hope, to build up a bit more neuroimmunology research in some of the centres where it's actually not being really supported at all in the past.
We are moving to the last couple of questions. What is your advice to a young medical student who is thinking of how to expand their career from medical school to postgraduate, so on and so forth?
Well, I think it's so different now that in a way, I think it's fairly obvious to most of them, at least in Oxford, but not in all medical schools in the UK by any means. In fact, rather few now. There is this opportunity. In fact, in Oxford, you have to do a 3rd year where there's much more science before you actually start the clinical aspects of your undergraduate career. And that's, I think, a very important time where they can experiment with their abilities to do experiments, if you like. So they do projects which are not very long, but they're long enough to get some really good results if they're well supervised. And it does give them a chance to know whether they actually like scientific bench type work. A few of them don't do that. They do more library projects or computer or something which is more clinical perhaps, but not lab based. I think that's a very important opportunity. When they get to the clinical years, of which we do 3, then I think, of course, they have all the wonderful opportunities of studying patients individually. And I'm a great believer in case reports.
Mm-hmm.
I know they're sometimes difficult to publish, but I think if you have an interesting case and if you can just add something a bit more scientific to it, maybe beg some laboratory to help by analyzing the patient's blood or doing something, then I think you should be able to get it published. And that can be a real step forward in a career. To have anything on your CV that's a publication is always good. But for me personally, those case reports or these cases, the individual cases, have often been the stimulus for some sort of breakthrough. Mm-hmm. So I do think they're very important, and we should never neglect the study of really interesting and specific patients.
With a curious mind, like Sherlock Holmes, like eyes and observation power, isn't it?
Yes.
I guess that would be relevant for young doctors and even the junior neurologists also. I take that your advice to all of them would be have a curious mind, have research, clinical, computer-based or laboratory-based, whichever the thing that you are comfortable as a different egg in your basket.
Yes, I think that's right. You have to find out what you're good at and do it as well as you can. That's actually my main advice to anybody.
Right.
Never trying to do something just because everybody else is doing it, and if you're not particularly good at it, but there will be something that you are good at, and if you can do that, it can be very fruitful.
Again, thank you very much for your time today. Before we finish this interview for World Federation Neurology YouTube channel, what is your final message for potential future winners for Angela Vincent Award at each World Congress from now on? The next one, I'm eagerly waiting to see who that person going to be and which part of the world. That would be in Rome in 2021.
Oh, yes, of course. Well, I just think, like everything else, people have to make the most of what they can do. And I know that there are real difficulties in some of those countries. And Dr. Sakadi's interview has shown how very limited the resources can be. And yet, with that kind of determination and with generosity of spirit, and I'm sure he spends a lot of his own time and possibly money on trying to develop the neurology service for epilepsy in his country, with a lot of commitment and real interest in the people, I think you should be able to achieve great things. And I think it's clear from what you've been telling me, Tissa, that once you get yourself known as somebody who wants to make progress, there will be people who can help from around the world.
100%. 100%. As I said to you off this video, I remember my first teacher in clinical neurology as a young medical student years ago, who was trained in UK, of course, and when he came back to Sri Lanka, as I heard, he managed to get a EEG machine donated by a gentleman called Harry Mianadi from Netherlands. He came back to Sri Lanka and he went on to train some volunteers and brought patients to his clinical school, took histories, and I read that paper as a young graduate from Sri Lanka those days. That is still one of the best done clinical epidemiological studies in epilepsy. So the Dr. Sakadi, I'm hoping, would take us a long way ahead in years to come with this award. In fact, when I interviewed him after winning this award, he told me how important this award for him and how much encouragement he had. And I also told him that World Federation Neurology is not a very rich society, but despite that, we have a lot of folks with the large hearts, and they would support him as best as they can.
Good. Wonderful.
Thank you very much again, and thank you for your time, and thank you for supporting us with the ongoing Angela Vincent Award. Have a wonderful day.
Thank you, and it's been a real pleasure to talk to you, and congratulations, Dr. Sakani.
Thank you very much.